In 2018, a peer-reviewed scientific paper made headlines around the world with a claim that sounded straight out of a science fiction movie. A group of 33 researchers suggested that the Cambrian explosion, a period of rapid evolutionary diversification on Earth, was triggered by extraterrestrial viruses. But the most shocking part of their hypothesis was their explanation for the octopus. They proposed that cryopreserved octopus eggs arrived on Earth via icy meteors. They argued that the biological complexity of the octopus, a creature with a massive nervous system, camera-like eyes, and the ability to rapidly camouflage itself, appeared too suddenly in the fossil record to be the result of standard Darwinian evolution. It is an incredibly tempting thought, looking at an animal that can taste with its skin, think with its arms, and edit its own genetic code on the fly. However, the scientific consensus strongly disagrees with the Meteor theory. The truth is much more grounded, yet infinitely more fascinating. The octopus is not an invader from the cosmos. It is the ultimate masterpiece of evolutionary adaptation right here in our own oceans. Their bizarre biology is not proof of extraterrestrial origins, but rather a testament to the extreme lengths life on our planet will go to survive. We do not need to look to the stars to find alien life. We just need to dive deep into the water. Today we will explore why octopuses seem like they do not belong on Earth, and the hardcore science behind their alien-like existence. If you enjoyed diving into the deep science of our natural world, please hit the like button and subscribe to the channel. Also, drop a comment below letting us know where in the world you're watching from and what time it is there right now. Let us begin uncovering the greatest biological mystery of the ocean. To truly comprehend the bizarre, almost extraterrestrial nature of the modern octopus, we must first travel back in deep time, rewinding the evolutionary clock by approximately 500 million years to the Cambrian and Ordovician periods. The ancient oceans were a radically different world, and the earliest ancestors of the octopus did not look anything like the soft, squishy, shape-shifting creatures we recognise today. They were heavily armoured, slow-moving biological tanks. These ancestral cephalopods were completely encased in rigid external shells, made primarily of a crystalline form of calcium carbonate known as aragonite. Imagine a creature resembling a modern chambered nautilus, but varying wildly in shape. Some possessed tightly coiled shells, while others, like the orthocones, had straight, massive conical shells that stretched up to several meters in length. During the Paleozoic era, these heavily armoured cephalopods were the undisputed apex predators of the marine ecosystem. They dominated the ancient seas, floating ominously through the water column with absolute impunity. The ocean was a different place back then. Predators were relatively slow and heavy. Thick armour was the ultimate foolproof defence mechanism against attacks. To understand why the octopus evolutionary lineage eventually decided to throw away this impenetrable fortress, we first need to dissect the immense biological, chemical and physical costs of carrying it around. Building and maintaining a shell is not biologically free. In fact, it requires an astronomical metabolic budget. The animal must constantly and actively extract dissolved calcium and carbonate ions from the surrounding seawater. It must then process these chemical compounds through a highly specialised layer of tissue called the mantle, and meticulously crystallise them into microscopic layers of aragonite, interspersed with a biological protein matrix for structural integrity. This continuous biomineralization process drains a massive amount of metabolic energy that could otherwise be allocated to somatic growth, reproductive success, or the development of a complex nervous system. Beyond the exhausting chemical cost of construction, there is the intense physical burden of the shell itself. Calcium carbonate is incredibly dense and heavy. For a soft-bodied animal to swim while dragging a massive stone house, it had to master complex fluid dynamics and buoyancy control. Ancestral cephalopods evolved a brilliant yet inherently limiting anatomical feature called a phragmocone. This was a series of internal walled-off chambers within the shell, connected by a living vascularized tube of tissue known as the sipuncle. The animal used the siphuncle to actively pump water out of the rear chambers through a process of osmosis, replacing the fluid with a precise mixture of nitrogen, oxygen, and carbon dioxide gases extracted from its own bloodstream. By meticulously adjusting the gas to liquid ratio within these chambers, the creature could achieve perfect neutral buoyancy, floating weightlessly in the water column like a meticulously engineered submarine. However, this physiological buoyancy system came with severe, fatal limitations. Gas-filled chambers are highly susceptible to extreme environmental pressure. If the shelled cephalopod dove too deep, the immense hydrostatic pressure of the deep ocean water would physically implode the shell, crushing the animal instantly in a violent collapse. Furthermore, a rigid gas-filled calcium carbonate cone is not exactly aerodynamic or hydrodynamically agile. These armoured giants could move forward and backward by expelling water through a funnel, utilising basic jet propulsion, but they were incredibly sluggish. They could not turn on a dime, they could not hide in narrow rocky crevices, and their overall acceleration was severely capped by the massive hydrodynamic drag of their own biological armour. For hundreds of millions of years, being a slow floating fortress was a highly successful survival strategy. But the planet Earth is not a static environment. Evolution is a perpetual arms race, and the rules of the biological game were about to change violently. As the timeline moved through the Devonian period and escalated into what paleontologists call the Mesozoic marine revolution, a terrifying new type of predator emerged on the evolutionary scene. This era saw the explosive rise of jawed fishes, teleost fishes, and later, incredibly fast-swimming marine reptiles like the ichthyosaurs and the plesiosaurs. These new oceanic predators possessed robust internal skeletal systems, highly efficient muscular arrangements, powerful tail fins for explosive, sustained speed, and devastating, bone-crushing jaws that could easily crack open a calcium carbonate shell like a brittle walnut. Suddenly, the slow-moving armoured cephalopods were sitting ducks. The oceanic paradigm had shifted completely. The very heavy shell that had successfully protected them for hundreds of millions of years became a fatal evolutionary trap. They could not outswim the new streamlined teleost fishes, and their rigid armour could no longer withstand the immense biting force of the massive marine reptiles. The entire cephalopod lineage faced a massive existential evolutionary bottleneck. The biological imperative was simple: adapt or face complete extinction. Under this immense predatory pressure, the lineage split. Some species maintained their shells and retreated to the deep, dark, less populated waters, surviving into the modern day as the Nautilus. But another group, driven by relentless natural selection, took a radical, counterintuitive evolutionary leap. Over millions of years, this diverging group, which would eventually become the subclass Coleoidea, began to internalize their external shells, shrinking them generation by microscopic generation. They essentially realized through natural selection that survival no longer depended on being an impenetrable fortress. Survival in the new, fast-paced ocean now depended entirely on speed, stealth, and unparalleled agility. The thick external shell first moved inside the mantle tissue, transforming into an internal buoyancy device, much like the porous cuttlebone found in modern cuttlefish, or the thin, transparent, flexible structure known as the gladius, or pen found in modern squids. But the specific ancestors of the octopus took this extreme biological reduction all the way to its absolute physiological limit. They completely eradicated the shell. They discarded the calcified remnants entirely, leaving the organism with absolutely no hard skeletal structures, neither internal nor external, with the sole exception of a sharp chitinous beak used for tearing prey. This total morphological reduction was a monumental evolutionary gamble. By willingly giving up their primary physical armour, they became arguably the most vulnerable creatures in the entire ocean. Practically every single predator loves the taste of unprotected, highly nutritious cephalopod protein. But this state of extreme physical vulnerability was the precise biological catalyst that forged their incredibly alien-like biology. Without the crushing metabolic burden of constantly secreting calcium carbonate, the ancestral octopus suddenly found itself with a massive surplus of biological energy. And without a rigid, inflexible shell restricting their physical growth and morphological shape, they could completely redesign their internal and external anatomy. Evolution aggressively redirected the metabolic energy once used for shell building into developing a highly sophisticated, hyperdense muscular architecture and a massive complex nervous system. Because they lacked bones, they evolved into what biomechanics experts classify as a muscular hydrostat. In a human body, or in the body of a predatory fish, muscles generate movement by pulling against rigid bones at hinged joints. But how does an organism move, grasp, or generate devastating mechanical force when it has absolutely no bones to act as levers? A muscular hydrostat relies on the fundamental physical principle that biological tissues are mostly composed of water, and water is essentially an incompressible fluid. The human tongue and the muscular trunk of an elephant are classic examples of muscular hydrostats, but the octopus is a living muscular hydrostat in its absolute entirety. The 8 arms of an octopus are densely packed with highly complex overlapping arrays of longitudinal, transverse, and helical muscle fibers. Because the overall volume of the arm must remain constant due to the incompressibility of the internal fluids, when one specific set of muscle fibers contracts and shortens, the tissue is physically forced to expand in a different direction. If the transverse muscles contract, reducing the diameter of the arm, the longitudinal muscles are forced to stretch, causing the arm to dramatically elongate. This constant dynamic interplay allows the octopus muscles to act as their own skeletal support system moment by moment. The resulting biomechanical system grants the organism infinite degrees of freedom. Unlike a jointed human arm that can only bend at the elbow and wrist, an octopus can bend any part of its arm, in any specific direction, at any mathematically possible angle, at any exact given moment in time. By sacrificing the rigid shell, the octopus gained the supernatural ability to manipulate its physical form entirely. It can compress its entire squishy, boneless body to squeeze through a tiny hole no larger than its single hard part, the beak between its eyes. This profound physical flexibility turned the rocky crevices, discarded shells, and narrow tunnels of the ocean floor into a new kind of impenetrable fortress, one that no jawed fish or marine reptile could ever physically reach. This singular, radical, evolutionary decision to undress, to completely abandon the heavy calcified armor of the deep past, was the critical turning point. It paved the direct way for the development of their massive, decentralized brains, their explosive burst speed, and their almost supernatural ability to completely disappear into the surrounding environment. They traded physical defense for pure, unadulterated biological intelligence and supreme physical adaptability, setting the stage for one of the most complex genetic blueprints on planet Earth. While their physical evolution from armoured tanks to soft-bodied escape artists is remarkable, the true alien nature of the octopus is hidden deep within its microscopic biology, specifically within its DNA. For decades, marine biologists and geneticists looked at the complex behaviour and advanced neurobiology of the octopus and assumed that their genetic code must be incredibly sophisticated. But it was not until 2015 that science finally got a comprehensive look under the hood. An international team of researchers achieved a monumental milestone. They fully sequenced the genome of the California two-spot octopus, scientifically known as Octopus bimaculoides. When the results were finally compiled and analysed, the scientific community was stunned. The genetic blueprint of the octopus was not just complex, it was massive, weird, and completely unlike anything they expected to find in a marine invertebrate. To understand just how bizarre the octopus genome is, we need to compare it to our own. The human genome, the complete set of DNA instructions that makes you who you are, contains roughly 3 billion base pairs and approximately 20,000 protein-coding genes. Before the 2015 study, scientists generally assumed that invertebrates, being evolutionarily older and theoretically simpler than mammals, would have significantly smaller and less complex genomes. The octopus shattered that assumption. The genome of the California two-spot octopus is massive, containing around 2.7 billion base pairs. That is almost as large as the human genome, but size is not everything. The true shock came when researchers counted the number of protein-coding genes, the specific sequences of DNA that actively produce the proteins responsible for building and operating the organism. The octopus genome contains approximately 33,000 protein-coding genes. Let that sink in for a moment. An octopus, a boneless creature living in a den on the ocean floor, has roughly 13,000 more active protein-coding genes than a human being. How did a mollusk end up with a genetic instruction manual more extensive than the creature currently building spaceships and splitting the atom? The answer lies not just in the number of genes, but in how those genes are organized and manipulated. When geneticists mapped the octopus genome, they discovered that it was incredibly chaotic. In most species, related genes are usually clustered together on the same chromosome, like chapters in a well-organized book. For example, genes responsible for producing specific digestive enzymes in humans are usually found grouped together. But in the octopus, these gene clusters have been violently blown apart and scattered randomly across the entire genome. It is as if someone took the instruction manual for building an octopus, put it through a paper shredder, and then glued the pieces back together in a completely random order. This extreme genetic scrambling is driven by a massive presence of something called transposable elements, or jumping genes. Discovered in the mid-20th century by geneticist Barbara McClintock, jumping genes are sequences of DNA that have the unique, somewhat terrifying ability to physically cut or copy themselves and paste themselves into entirely new locations within the genome. In humans and most other vertebrates, jumping genes make up a significant portion of our DNA, but the vast majority of them are dormant. They have been mutated or silenced by evolutionary defense mechanisms over millions of years, turning them into genetic fossils, essentially junk DNA that no longer functions. The octopus, however, is a very different story. The octopus genome is absolutely crawling with highly active jumping genes, specifically a type known as transposons. These transposons are not dormant fossils. They're actively moving, copying and pasting themselves throughout the octopus's DNA at an astonishing rate. This constant genetic reshuffling is what caused the massive expansion of their genome and the chaotic scattering of their gene clusters. While this level of genetic instability would be highly detrimental or even fatal to most organisms, causing devastating mutations and cancers, the octopus has somehow harnessed this chaos. The rampant activity of jumping genes acts as a powerful engine for rapid evolutionary innovation. When a transposon jumps to a new location, it can alter the function of nearby genes, turn genes on or off, or even duplicate entire gene sequences. Over millions of years, this constant reshuffling allowed the octopus to experiment with new genetic combinations at a furious pace. This is likely how they rapidly expanded families of specific genes, such as the zinc finger transcription factors, which are crucial for embryonic development and the formation of complex nervous systems. The octopus has hundreds of these genes, far more than most other invertebrates, and their rapid expansion was almost certainly driven by the relentless cut-and-paste action of their jumping genes. The discovery of the massive transposon-riddled octopus genome fundamentally changed our understanding of cephalopod evolution. It proved that their incredible biological complexity, their massive brains, their camera-like eyes, their shape-shifting abilities, was not a sudden magical occurrence. nor was it a gift from a passing meteor. It was the result of a highly dynamic, inherently unstable genome that constantly experimented, duplicated, and reshuffled its own instructions until it stumbled upon the perfect biological design for surviving in a hostile ocean without a shell. They are not aliens, they are the ultimate genetic gamblers, and they hit the evolutionary jackpot. While the massive size of their genome and the chaotic frenzy of their jumping genes are undeniably mind-bending, these factors alone do not fully explain the octopus's supernatural ability to survive in radically different oceanic environments. To truly grasp the alien nature of their biology, we have to look at how they fundamentally break the most sacred rule of modern biology. In the scientific community, there is a foundational principle known as the central dogma of molecular biology. This dogma dictates the strict, unidirectional flow of genetic information within a living organism. It states that instructions are hard-coded in the deoxyribonucleic acid, which we commonly call DNA. When a cell needs to build a protein, it copies a specific section of that DNA into a temporary messenger molecule called messenger ribonucleic acid. This messenger, ribonucleic acid, then travels to the protein-building factories of the cell, where its sequence is read to assemble the final protein. Under this traditional biological framework, the DNA is the absolute master blueprint. The messenger ribonucleic acid is merely a passive courier, and the resulting protein is the physical expression of that exact unchanged genetic code. If an organism wants to change its physical traits or adapt to a new environment, it must wait for random genetic mutations to occur in its DNA. This process of Darwinian evolution is agonizingly slow. It takes thousands, if not millions of years, and countless generations of natural selection to permanently alter the DNA blueprint and produce a beneficial new protein. For a creature like a human, or a whale, or a shark, this slow evolutionary timeline works perfectly fine. But for the octopus, waiting 1,000,000 years for a genetic mutation is simply not an option. Remember, they sacrifice their protective shells to become soft-bodied masters of movement. Because of this extreme mobility, a single octopus might hatch in the warm, shallow, sunlit waters of a tropical reef, and then, mere weeks later, migrate down into the crushing depths of the deep ocean trench, where the environment is pitch black and the water temperature plummets to near freezing. If an octopus relied strictly on the slow mutation of its DNA to adapt to these brutal temperature shifts, its nervous system would shut down, and it would die in a matter of hours. The environmental pressure is immediate, but genetic evolution is generational. To solve this seemingly impossible biological paradox, the octopus evolved the most sophisticated, jaw-dropping molecular hack ever discovered in the animal kingdom. They learned how to intercept and rewrite their own genetic code on the fly. They do this through a highly active process known as messenger ribonucleic acid editing. Instead of waiting for their DNA to mutate over millions of years, the octopus actively edits the messenger molecules after they have been copied from the DNA, but before they reach the protein-building factories. It is exactly like an architect sending a printed blueprint to a construction site. But a rogue engineer intercepts the courier in the hallway, crosses out critical measurements with a red pen, writes in new instructions, and then sends the altered blueprint to the builders. The original blueprint sitting in the office remains completely unchanged, but the building that actually gets constructed is entirely different. How does the octopus perform this biological sorcery? They utilize a highly specialized family of enzymes called adenosine deaminases, acting on ribonucleic acid. In the language of genetics, the code is written using 4 chemical bases: adenosine, cytosine, guanosine, and uracil. When the octopus needs to adapt to a sudden environmental change, these specialized enzymes chemically bind to the messenger ribonucleic acid strand. The enzyme targets a specific adenosine molecule and physically rips an amino group away from it, replacing it with an oxygen atom from a surrounding water molecule. This incredibly precise chemical reaction transforms the adenosine into an entirely different molecule called inosine. This might sound like a microscopic, insignificant detail, but it has catastrophic, beautiful implications for the organism. When the cell's protein-building machinery eventually reads this newly edited sequence, it does not recognize the inosine. Instead, it reads the inosine as if it were a guanosine molecule. By changing a single molecular letter in the messenger sequence, the octopus fundamentally alters the instructions for building the protein. The final protein that gets manufactured will have a different sequence of amino acids, which changes its three-dimensional physical shape and consequently changes its entire biological function. To understand just how insanely powerful this is, we have to look at the octopus nervous system in cold water. Like humans, an octopus's nervous system relies on electrical signals to think, move, and react. These electrical signals are generated by specialized proteins called ion channels, specifically potassium and sodium channels, which sit on the surface of nerve cells. These channels physically open and close to let charged ions flow in and out of the nerve cell, firing electrical impulses. The speed at which these protein channels open and close is highly dependent on temperature. In warm water, they fire rapidly. But when an octopus dives into freezing deep water, thermodynamics takes over. The cold temperature causes the lipid membranes of the nerve cells to stiffen, and the potassium channel proteins become sluggish, opening and closing far too slowly. If this were to happen to a human, our nervous system would crash, resulting in hypothermia, paralysis, and death. When the octopus enters cold water, it detects the temperature drop and immediately ramps up its ribonucleic acid editing machinery. The enzymes swarm the messenger molecules that are carrying the instructions for building potassium channels. They edit the adenosine molecules into inosine molecules at incredibly specific locations. The edited messenger molecules are then used to build new potassium channels. Because of the edits, these new proteins have a slightly different physical structure. This new modified shape prevents them from becoming sluggish in the cold. They are essentially specialized, cold-weather proteins manufactured on demand. Within hours, the octopus replaces the standard potassium channels in its nervous system with these freshly edited, cold-resistant versions. Its brain and tentacles continue to fire electrical signals at maximum speed, completely ignoring the freezing temperatures outside. When the octopus swims back up to the warm surface waters, it simply stops editing those specific ribonucleic acid messengers, and the body reverts to building the standard, warm weather proteins. They are essentially shape-shifting at the molecular level, adapting their biochemistry in real time to suit whatever environment they wander into. While humans and other mammals do possess the ability to edit ribonucleic acid, we use it very sparingly. In humans, significant ribonucleic acid editing only occurs in a tiny fraction of our genes, and it is usually restricted to minor physiological tuning or disease prevention. The octopus, however, uses this mechanism on an industrial scale. Researchers have discovered that octopuses, squids, and cuttlefish edit their ribonucleic acid at tens of thousands of different sites, particularly in the genes related to their nervous systems. More than 60% of the ribonucleic acid transcripts in an octopus brain are actively edited, a volume of genetic manipulation that is absolutely unprecedented in the natural world. However, this incredible superpower comes with a massive evolutionary trade-off. In order for the editing enzymes to recognize exactly where to make their cuts, the genetic sequence surrounding the target site must remain perfectly stable and highly structured. If the underlying DNA mutates even slightly, the complex folding of the messenger molecule will change, the enzyme will not be able to bind to it, and the entire editing system will collapse. Because the octopus relies so heavily on ribonucleic acid editing for its daily survival, it cannot afford to let its DNA mutate. Natural selection fiercely eliminates any octopus that develops mutations in these critical areas. Therefore, by choosing to master the art of instant molecular adaptation, the octopus essentially hit the pause button on its own long-term genomic evolution. Their DNA evolves at a vastly slower rate than almost any other animal on the planet. They sacrificed the long-term, slow-burn potential of Darwinian DNA mutation for the immediate short-term superpower of physiological flexibility. They are an evolutionary paradox. Their bodies and brains are capable of the most complex rapid adaptations on Earth, yet their foundational genetic code is locked in a state of extreme, conservative stasis. This is why their biology feels so alien. They did not just adapt to their environment, they found a loophole in the fundamental laws of genetics and exploited it to absolute perfection, bypassing the slow march of evolution to become masters of their own immediate destiny. As human beings, our entire conceptual understanding of intelligence is heavily biased by our own anatomy. We operate on a strictly centralized biological architecture. In vertebrates, the nervous system is designed like a highly secure, top-down corporate hierarchy. The brain acts as the undisputed chief executive officer, locked safely inside a heavily armoured vault of bone at the top of the body, while the spinal cord acts as the main transmission cable, dictating every single muscular movement and processing all sensory data. To comprehend the neurobiology of the octopus, we must completely shatter this vertebrate blueprint and throw it away. The octopus has evolved a neurological architecture so radically decentralized and profoundly alien that marine biologists frequently refer to them as having 9 separate brains. This is not just a poetic metaphor. It is an incredibly accurate description of how their neurological network physically and mechanically operates in the ocean. To begin mapping this bizarre intelligence, we first need to look at the sheer processing power the animal possesses. An average mature octopus boasts a nervous system containing roughly 500 million individual neurons. In the biological hierarchy of the animal kingdom, this is a staggering, almost unbelievable number for an invertebrate. It completely eclipses the neural density of insects, spiders, or even other mollusks. 500 million neurons place the octopus in the same intellectual weight class as a golden retriever or a domestic cat. However, it is not the raw number of neurons that makes them seem like extraterrestrials. It is exactly where those neurons are physically located and how they communicate with one another. If you were to look for a traditional centralized brain in an octopus, you would be deeply confused. Only about 1/3 of those 500 million neurons, roughly 170 million of them, reside in what we would call the central brain located inside the animal's head. Even this central processing unit is a masterpiece of biological absurdity. Because the octopus entirely lacks a skeletal system, its central brain is not protected by a skull. Instead, it is housed within a flexible, cartilaginous capsule. But the most horrifying and fascinating aspect of its anatomical placement is its shape. The central brain of an octopus is shaped exactly like a biological donut. It forms a thick neural ring, and running directly through the exact centre of this brain ring is the animal's esophagus, the primary feeding tube connecting the mouth to the stomach. This creates a terrifying anatomical hazard. If an octopus is too greedy and attempts to swallow a piece of a crustacean shell or a fishbone that is slightly too large, the sharp, rigid object passing through the esophagus could bulge outward and physically scrape, compress, or severely lacerate the inner walls of its own brain. To prevent fatal brain damage every time it eats, the octopus is strictly forced to use its sharp, chitinous beak to meticulously tear its prey into microscopic, heavily pulverized shreds before swallowing. Despite this precarious placement, the central brain is incredibly advanced. It is divided into dozens of highly specialized lobes. The largest of these are the optic lobes, which process the massive influx of visual data coming from their highly complex camera-like eyes. Behind these lies the vertical lobe, a dense cluster of neurons dedicated almost entirely to advanced learning, spatial navigation, and long-term memory storage. The central brain is capable of tremendous cognitive feats, but it does not micromanage the animal's body. It cannot, because it simply does not possess enough neurons to do so. This brings us to the true marvel of cephalopod neurobiology. The remaining 2/3 of the animal's neurons, over 330 million independent nerve cells, are not in the head at all. They are distributed outward, heavily packed into the 8 muscular arms. Each individual arm contains a massive, incredibly thick nerve cord running directly down its centre. This nerve cord is not just a dumb biological wire, blindly transmitting signals back to the central brain, like the nerves in a human arm. Instead, it is a highly sophisticated, densely interwoven chain of neural ganglia. These ganglia act as independent processing centres, containing vast networks of interneurons and complex reflex loops. From a functional neurobiological standpoint, each of the 8 arms possesses its own localized autonomous brain. This distributed neural network fundamentally changes how the organism interacts with the physical universe. In a human, if your finger brushes against a sharp object, sensory neurons send a frantic electrical signal all the way up your arm, into your spinal cord, and up to your brain. Your brain processes the pain, formulates a motor response, and sends a signal all the way back down the arm to pull your hand away. In an octopus, this centralized feedback loop is completely bypassed. The suckers lining each arm are packed with tens of thousands of specialized mechanoreceptors for feeling texture and extreme physical pressure, as well as highly sensitive chemoreceptors that allow the octopus to physically taste whatever it touches. When an octopus arm is sliding blindly into a dark, rocky crevice on the ocean floor, and one of its suckers tastes the chemical signature of a hidden crab, the localized arm brain processes this sensory data immediately. The arm makes its own independent, calculated decision to stretch, fire its muscles, and tightly wrap around the prey, all without ever bothering to send the sensory data up to the central brain for approval. The question then arises, if an animal has 8 independent thinking arms and 1 central executive brain, how does it not descend into absolute neurological chaos? How does an octopus keep its arms from fighting each other, or tying themselves into permanent catastrophic knots? The answer lies in a brilliant system of hierarchical command and lateral communication. The entire nervous system operates on a principle of delegated authority. The central brain acts as a high-level strategic commander. It processes the visual field, evaluates the overall environment, and broadcasts a generalized abstract command. The central brain basically transmits a signal that says, "We are hungry, and there is a crab hiding behind that rock. Arms, go get it." The central executive brain does not calculate the specific muscle contractions required to reach the crab. It simply issues the goal. The autonomous nerve cords in the arms receive this high-level directive and take over the complex mathematical execution. Furthermore, the arms do not operate in complete isolation. At the very base of the 8 arms, just below the central brain, there is a circular neural highway known as the brachial ring. This ring acts as a massive high-speed data cable, connecting the nerve cords of all 8 arms directly to one another. This lateral connection allows the arm brains to continuously share sensory data, spatial coordinates, and physical intent with their neighbouring arms, completely bypassing the central brain. If arm number 1 decides to anchor itself to a rock, it instantly communicates its exact physical coordinates to arm number 2 through the brachial ring, ensuring they work in synchronized fluid harmony rather than tangled opposition. What makes this completely alien, decentralized architecture even more shocking is the chemical foundation upon which it is built. You might assume that such a bizarre, non-vertebrate brain would require completely alien chemistry to function. Yet, neurobiologists have discovered that the octopus nervous system is powered by the exact same chemical messengers, the precise neurotransmitters that exist in the human brain. When an octopus is learning to solve a complex puzzle, its neurons are firing using glutamate and acetylcholine. When they are regulating their mood, aggression, or complex social behaviors, their nervous systems are flooded with serotonin. and when an octopus successfully captures a difficult prey or mates, its brain releases dopamine, the exact same molecule responsible for the sensation of reward and pleasure in human beings. This is a profound example of convergent evolution at the deepest, most microscopic chemical level. The biological hardware is radically different, but the chemical software running the machine is eerily familiar. The ultimate reason for this extreme decentralization comes down to pure physics and biomechanical necessity. As we discussed in their evolutionary history, the octopus traded a rigid skeleton for a body of pure, infinite flexibility. A vertebrate animal with a fixed skeleton only needs its central brain to calculate a severely limited number of joint angles to coordinate movement. An elbow can only bend in one specific direction, but an octopus arm, being a muscular hydrostat, has virtually infinite degrees of mechanical freedom. It can bend, twist, stretch, or compress at any exact mathematical point along its entire length, in three-dimensional space, at any given millisecond. If the central brain were forced to consciously calculate and orchestrate the exact micro-muscular contractions required to coordinate all 8 arms simultaneously, the mathematical processing power required would be astronomical. The central brain would need to be the size of a small car, demanding an impossible biological supply of oxygen and glucose to survive. By offloading the vast majority of the sensory processing and motor control directly into the limbs, the octopus solved an impossible computational physics problem. It distributed the processing power exactly where it is needed most, at the point of physical contact with the environment. They did not just evolve a brain, they evolved a living, thinking, decentralized neural web, transforming their entire body into an organ of pure cognition. To fully comprehend the terrifying and beautiful reality of a decentralized nervous system, we must look at what happens when the physical connection between the central brain and the limbs is violently severed. In the realm of mammalian biology, if an arm is amputated, the limb is instantly rendered inert. It becomes lifeless flesh because the solitary command center, the brain, has been disconnected. But the octopus operates under a completely different set of biological laws. In scientific laboratories, neurobiologists have conducted observations on severed octopus arms that sound like they belong in a horror film, yet they perfectly illustrate the profound independence of these appendages. When an octopus arm is surgically removed from the body, it does not simply die or twitch randomly. Because it contains its own localized brain, complete with millions of neurons and intact reflex loops, the severed arm remains highly active and seemingly aware for an astonishingly long time. If you place a living crab next to a recently severed octopus arm, the arm will detect the chemical signature of the prey. The autonomous nerve cord inside the arm will process this sensory data, fire the complex muscular hydrostat system, and the arm will physically crawl toward the crab. These suckers will independently coordinate to grasp the prey, subdue it, and then, in a display of phantom biological programming, the arm will bend backward, attempting to pass the captured food to a mouth that is no longer there. The severed limb retains its basic localized programming: hunt, grab, and feed. This is not a mere muscle spasm, it is a complex, multi-step behavioral sequence executed entirely by the neural architecture embedded within the arm itself, completely isolated from any central processing unit. This extreme autonomy solves a massive computational problem for the octopus, specifically the problem of proprioception. In the human body, proprioception is our internal three-dimensional sense of space. Even with your eyes closed, your central brain knows exactly where your hands and feet are, thanks to thousands of specialized stretch receptors in your muscles and joints constantly sending spatial coordinates back to the brain. The brain continuously updates a strict topographical map of the entire body, but an octopus has no fixed joints and no skeletal structure. Its 8 arms can twist, stretch, and deform in infinite mathematical variations. If the central brain attempted to track the exact physical configuration of all 8 arms at every given millisecond, the mathematical data load would cause a catastrophic neural crash, Evolution solved this by simply eliminating central proprioception. The central brain of the octopus is remarkably blind to its own body. The high-level commander in the head does not know the exact shape or position of its arms. When the central brain wants to move an arm to a specific location, it does not send a detailed coordinate-based command. Instead, it sends a generalized command down the nerve cord. The autonomous arm then relies entirely on its own local sensory input to calculate the trajectory and execute the movement. The arms are essentially operating on autopilot, exploring the environment, feeling their way around obstacles, and making micro-adjustments without ever bothering the central brain with the trivial details of their physical geometry. The primary tools the arms use to navigate and interact with the world are the suckers, which are arguably the most sophisticated sensory organs in the invertebrate kingdom. An average octopus possesses roughly 2,000 suckers distributed across its 8 arms, and each individual sucker is a highly complex sensory processing unit. They are packed with tens of thousands of specialized receptors that grant the octopus a supernatural sensory crossover, the ability to taste by touch. While human taste buds are confined to the tongue and require molecules to be dissolved in liquid, the chemotactile receptors on an octopus sucker are designed to detect poorly soluble molecules directly upon physical contact. When an octopus grazes its arm over a seemingly barren rock, its suckers are essentially licking the surface, instantly identifying the chemical residue of a snail that passed by hours ago, or detecting the hidden proteins of a camouflaged crab buried in the sand. With 8 autonomous arms constantly tasting, feeling, and reacting to the environment, all while the central brain is largely unaware of their exact physical coordinates. One might wonder why the octopus does not accidentally hunt itself. Why do the hundreds of independently grasping suckers not grab onto the animal's other arms, tying the creature into a permanent fatal knot? The answer lies in a brilliant localized chemical defense mechanism. The skin of the octopus secretes a specific, specialized chemical marker. The suckers on the arms are hardwired at the local neural level to instantly recognize this chemical signature. When a sucker comes into contact with octopus skin, the local nerve cord forcefully overrides the grabbing reflex. This self-recognition mechanism is so powerful that even a severed arm operating entirely on its own will refuse to grab an object coated in octopus skin extract. The arms recognize their own chemical self, ensuring that the decentralized network operates in fluid harmony rather than cannibalistic chaos. When we step back and analyze this bizarre distributed intelligence, we are forced to confront one of the most profound and philosophically staggering concepts in evolutionary biology, the phenomenon of convergent evolution. To understand the magnitude of this concept, we have to look at the evolutionary family tree of life on Earth. Humans and octopuses do share a common ancestor, but you have to travel back in deep time, roughly 600 million years, to find it. This last common ancestor was a microscopic, worm-like creature crawling in the primordial mud of the ancient oceans. It had no brain, no complex eyes, and no articulated limbs. It was little more than a biological tube designed to process nutrients. From that humble, brainless worm, the evolutionary tree violently split. One massive branch ventured down the path of the chordates, leading to vertebrates, fish, reptiles, mammals, and eventually human beings. The other branch veered off into the invertebrates, leading to mollusks, snails, clams, and eventually the cephalopods. For 600 million years, these 2 genetic lineages evolved in completely total isolation from one another. They faced different environments, different predators, and different physical constraints. Yet despite this massive evolutionary chasm, both lineages eventually arrived at the exact same biological destination: high-level, complex intelligence. This means that the universe, or at least the biological ecosystem of planet Earth, essentially ran 2 completely independent experiments on how to build a sentient thinking mind. The vertebrate path centralized the processing power, encased it in bone, and developed highly specialized insulating tissues like myelin sheaths around the nerve fibers to increase the speed of electrical transmission. The invertebrate cephalopod path threw all those blueprints out the window. Because they did not evolve myelin to insulate their nerves, the octopus had to find a different physical solution to transmit electrical signals quickly. They achieved this by evolving giant axons, massively thick nerve fibers that compensate for the lack of insulation through sheer physical volume. They decentralized the processing power, scattered it throughout their soft limbs, and abandoned the central bodily map. If human intelligence is akin to a highly centralized mainframe computer system running a specific operating system, then octopus intelligence is a radically decentralized blockchain-like network running on entirely different source code. Yet, both biological machines are capable of incredibly complex outputs. They both experience curiosity, they both navigate complex spatial mazes, they both learn from their physical environments, and they both possess the capacity to adapt to new, unexpected problems. Convergent evolution proves that intelligence, consciousness, and complex problem-solving are not evolutionary flukes unique to the human lineage. They are highly advantageous biological endpoints that natural selection will relentlessly drive toward, even if it has to invent entirely new architectural blueprints to get there. The octopus forces us to redefine what a brain actually is. We are so accustomed to thinking of the mind as a singular, isolated entity sitting in a dark room inside our skulls, piloting a dumb biological vehicle. But the octopus is not a passenger in its body. It is a creature whose very flesh is infused with cognition. Its mind is not confined to its head. It extends all the way to the tips of its 8 arms. Every sucker, every muscle fiber, and every decentralized ganglion is a functioning part of its conscious experience. By studying the octopus, we are not just analysing a weird marine mollusk. We are literally studying an alternate genesis of intelligence. We are looking at a creature that achieved a mammalian level of cognitive complexity using an entirely different set of biological Lego bricks, proving that the emergence of a complex mind in the universe is far more inevitable and far more diverse than we ever dared to imagine. When evolutionary biologists and physicists observe an octopus hunting or hiding on a coral reef, they are witnessing what is arguably the most advanced optical illusion in the entire animal kingdom. The octopus possesses the supernatural ability to render itself practically invisible to both predators and prey in a matter of milliseconds. This is not a gradual colour shift, like a chameleon taking several minutes to adapt to a branch. This is instantaneous, high-resolution, real-time camouflage. To understand how they achieve this optical sorcery, we must peel back the layers of their skin and examine the microscopic physics of their bioluminescence and pigmentation. The skin of an octopus is not just a passive covering. It is a hyperactive, neurologically driven video display screen. The primary engine of this visual display is a massive network of specialized cells known as chromatophores. An adult octopus can possess millions of these microscopic pigment sacs embedded in the upper layers of its skin. However, these are not standard pigment cells like the melanocytes that determine human skin color. A cephalopod chromatophore is a complex biomechanical organ. At the center of each chromatophore is an elastic sac known as a cytoelastic sacculus, which is completely filled with a dense, brightly colored pigment granule. Surrounding this central sac is a precise radial array of 15 to 25 microscopic muscle fibers. These muscles are directly innervated by the autonomous nerve cords running through the animal's limbs and connected to the central brain. This direct neurological connection is the key to their terrifying speed. When an octopus wants to change its colour, the brain fires an electrical signal down to the skin. The microscopic muscles attached to a specific chromatophore violently contract, pulling the elastic sac outward in all directions. As the sac stretches, the concentrated pigment inside is spread out, flattening into a wide, visible disc of colour on the surface of the skin. When the electrical signal ceases, the muscles relax, the elastic sac violently snaps back to its original microscopic size, and the colour instantly vanishes. By precisely controlling which muscle fibres contract and relax, the octopus can rapidly display complex patterns of yellow, orange, red, brown, and black. However, manipulating pigment is only the first layer of the illusion. If an octopus only had chromatophores, it could only mimic dark or warm coloured environments. To truly disappear into the shifting blues, greens, and brilliant whites of a sunlit coral reef, the octopus must manipulate the fundamental physics of light itself. Below the layer of chromatophores lies a second, highly specialised layer of cells called iridophores. Unlike chromatophores, which use pigment to absorb certain wavelengths of light and reflect others, iridophores do not contain any pigment at all. They operate entirely on the principles of structural colour and thin film interference. Inside an iridophore cell are highly organised stacks of ultra-thin protein plates called reflectins. These plates are separated by microscopic layers of cytoplasm. When ambient light from the ocean surface penetrates the octopus's skin and hits these protein stacks, a complex physical interaction occurs. Some of the light reflects off the top surface of the first protein plate, while some of the light penetrates deeper, reflecting off the second, third, or fourth plates. When these multiple reflected light waves travel back out of the cell, they physically interfere with one another. Depending on the exact distance between the protein plates, certain wavelengths of light will amplify each other, constructive interference, while other wavelengths will cancel each other out, destructive interference. By neurologically altering the physical spacing between these microscopic reflectin plates, the octopus can actively tune the exact wavelength of light that the iridophore reflects. They can shift the structural reflection from a deep, iridescent blue to a vibrant emerald green in an instant. This provides the crucial cold colours necessary to blend into the oceanic background. But the optical stack does not stop there. Beneath the iridophores lies a third, deeply buried layer of cells known as leucophores. Leucophores are the ultimate background manipulators. They are flattened cells filled with highly reflective crystalline purines. Their sole physical purpose is to act as a broadband, biological mirror. They do not manipulate specific wavelengths or create structural interference. Instead, they catch whatever ambient light is available in the surrounding environment and perfectly scatter it back through the upper layers of the skin. If an octopus is hiding in a dark, muddy crevice, the leucophores reflect the dark ambient light. If the octopus is swimming through a bright sunlit patch of white sand, the leucophores scatter the brilliant white light. They provide the perfect foundational canvas upon which the chromatophores and iridophores can paint their active illusions. The coordination of this three-tiered optical system is a masterpiece of biological engineering. When an octopus settles onto a complex background, say, a patch of algae-covered rock sitting on white sand, the localized brains in its arms analyze the visual texture. The autonomous nervous system instantly calculates the precise combination of electrical signals required. It fires specific chromatophore muscles to create brown and red speckles mimicking the algae. It tunes the iridophores to reflect the subtle green hues of the plant life, and it relies on the deep leucophores to bounce back the ambient white light of the surrounding sand. The result is a hyper-realistic, three-dimensional biological rendering of the environment. But true camouflage is not just about colour, it is also about texture. A perfectly coloured octopus will still cast a shadow and look suspicious if its skin is completely smooth while resting on a jagged rock. To solve this physical geometry problem, the octopus utilises a fourth layer of biomechanical mastery, papillae. Scattered across the surface of the skin are complex networks of highly specialised erector sussels. These muscles can instantly force the skin to physically bulge, spike, or fold, altering its three-dimensional topography. By firing these papillae muscles in specific combinations, The octopus can change its skin texture from perfectly smooth glass to jagged coral, bumpy gravel, or even stringy seaweed. When you combine the high-speed, neurologically driven colour shifting of the chromatophores, the structural light manipulation of the iridophores, the ambient mirroring of the leucophores, and the three-dimensional physical morphing of the papillae, you get an organism that can literally become its environment. They can mimic a venomous sea snake to deter predators. They can flatten out and glide across the sand mimicking a harmless flounder, or they can simply cease to exist visually, vanishing completely into a rock face. They have mastered the physics of light, colour and texture so completely that they have turned their own flesh into the ultimate optical weapon, proving once again that their evolutionary path is one of the most brilliant and terrifying designs in the natural world. We have just established that the octopus is the undisputed master of visual deception, capable of rendering itself invisible by manipulating microscopic colour pixels and structural reflectors across its entire body. However, the scientific explanation of chromatophores and iridophores immediately collides with a massive, seemingly impossible biological paradox. If you ask any marine biologist about the visual capabilities of an octopus, they will tell you a shocking fact. Octopuses are completely colourblind. How can an organism perfectly replicate the exact colours of a complex, vibrant coral reef when it fundamentally lacks the biological hardware to see those colours in the first place? To unravel this mind-bending mystery, we have to examine both the incredible physics of their specialised eyes and a deeply hidden, totally alien sensory system that operates entirely outside of their brain. First, let us look at the eyes themselves. The eyes of an octopus are gigantic relative to their body size, and they represent another stunning example of convergent evolution. Structurally, an octopus eye is remarkably similar to a human eye. It possesses a clear outer cornea, an adjustable iris, a focusing lens, and a dense retina at the back to capture light. But at the microscopic level, their retinas are fundamentally different from ours. Human retinas contain 3 distinct types of photoreceptor cells called cones, which are individually tuned to detect red, green, and blue wavelengths of light. By combining the data from these 3 cones, our brains compute the full spectrum of colour. The octopus retina, however, only contains a single type of photoreceptor. In the strict biological definition, having only one type of photoreceptor means the animal only sees the world in variations of grayscale. For decades, the scientific community was completely baffled by this. The octopus was executing high definition, perfectly colour-matched camouflage in a world it theoretically could only see in black and white. Initially, physicists proposed a brilliant, purely optical theory to explain this paradox. They pointed to the bizarre shape of the octopus pupil. While a human pupil is perfectly round, an octopus pupil is shaped like a wide horizontal letter U or a crescent moon. Physicists suggested that this extreme shape allows the octopus to exploit a physical phenomenon known as chromatic aberration. When light passes through a lens, different wavelengths, meaning different colours, bend at slightly different angles. Blue light bends more sharply than red light. In a camera or a human eye, this chromatic aberration is considered a flaw, and our biological lenses are designed to minimise it so we see a sharp image. But the octopus, with its massive U-shaped pupil, might actually maximise this effect. By rapidly shifting the focal depth of its eye, bringing the lens closer to or further from the retina, the octopus could theoretically force different wavelengths of light to snap in and out of focus one at a time. The brain could then mathematically calculate the colour of an object based entirely on the specific focal distance required to make its edges appear sharp. It is an ingenious theory, essentially suggesting they calculate colour using geometry and physical optics, rather than chemical photoreceptors. While chromatic aberration likely plays a role in their overall vision, marine biologists recently realised it was not the whole story. The speed and localised precision of their camouflage was simply too fast for the central brain to be constantly calculating focal geometries and sending commands back to the skin. The true answer, discovered in recent years, is far more science fiction than anyone anticipated. The octopus does not just see with its eyes. Its skin can actually see. In 2015, molecular biologists ran genetic and chemical analyses on the skin tissue of octopuses and cuttlefish, and they found massive concentrations of a specific protein called opsin. Opsins are the fundamental light-sensitive proteins that make vision possible in almost all complex animals. In human beings, opsins are strictly confined to the photoreceptor cells locked safely inside the retinas of our eyes. But in the octopus, these light-sensitive opsin proteins are distributed directly throughout the outer layers of their skin, woven directly into the vast network of chromatophores and iridophores. The octopus is literally covered in a decentralized biological retina. To understand how this skin vision operates, we have to look at the elegant biochemistry of the opsin molecule. Within the skin, the opsin protein is chemically bound to a smaller molecule called a chromophore, specifically a derivative of vitamin A known as retinol. This retinol molecule has a very specific bent physical shape. When a photon of light from the surrounding ocean strikes the octopus skin and hits this molecule, the pure energy of the photon causes the retinol to physically snap into a straight line. This microscopic, physical shape shifting from a bent isomer to a straight isomer is the foundational spark of all vision. When the retinol molecule straightens out, it violently pushes against the opsin protein it is attached to, changing the opsin's shape as well. This structural change activates the opsin, turning it into a biological switch that triggers a massive localized chemical cascade. The activated opsin interacts with nearby G proteins, which then activate a series of enzymes. These enzymes rapidly break down specific signaling molecules within the cell, causing specialized ion channels in the cell membrane to fly open. Calcium ions flood into the cell, creating an electrical current. In a human eye, this electrical current would travel down the optic nerve to the brain. But in the octopus skin, the architecture is radically different. The electrical signals generated by the light-sensitive opsins in the skin do not have to travel all the way up to the central donut-shaped brain to be processed. Instead, these signals are routed directly to the autonomous, localized nerve networks embedded within the arms and the mantle. This creates an incredibly fast, highly localized feedback loop. The skin physically detects the ambient wavelength of light hitting it, generates an electrical signal, and directly commands the adjacent chromatophore muscles to expand or contract, and directly alters the chemical hydration of the reflectin proteins within the iridophores to tune their iridescence. This means that a significant portion of an octopus's camouflage is entirely automated and decentralized. The central executive brain might send a general command that says, "We're exposed, initiate camouflage," but it is the skin itself acting as a massive distributed sensory organ that handles the fine pixel-by-pixel tuning. The skin on the left side of the arm might be detecting the blue wavelengths of open water and automatically firing the iridophores to reflect blue, while the skin on the right side of the exact same arm might be resting against red coral, detecting the red wavelengths and automatically firing the red chromatophores. The localized skin networks are making independent optical decisions based on direct photon reception, entirely bypassing the central processing unit. This concept of dermal photoreception completely shatters our conventional understanding of how sensory organs are supposed to work. For a vertebrate, the boundary between the organism and the environment is highly distinct. The skin is a passive barrier, a biological wall protecting the internal organs. We collect visual data through two highly specialized centralized windows, our eyes, and process that data in a centralized vault, our brain. The octopus, however, blurs the line between organism and environment to an almost philosophical degree. By pushing its visual processing power out to the very edges of its anatomy, the octopus transforms its entire body surface into a massive, active, thinking lens. When it moves across the ocean floor, its skin is constantly absorbing, analysing and reacting to the surrounding electromagnetic radiation. It is essentially tasting the ambient light. This decentralized visual system works in perfect synchronized harmony with their decentralized tactile system, where their suckers are constantly tasting and feeling the physical terrain. Through this dual mechanism of chromatic aberration in their massive eyes and decentralized opsin networks embedded directly in their skin, the octopus solve the colorblind paradox. They abandon the need for complex multi-channel color processing in the central brain, a process that would require a massive amount of metabolic energy and neural density. Instead, they opted for an immediate chemically driven automated response at the exact point of contact with the environment. Their skin is not just a canvas, it is a hyper-intelligent light-sensing matrix. They do not just wear their camouflage, they literally see through it. This bizarre, decentralized visual architecture is yet another piece of hardcore biological evidence, proving that the octopus operates on a completely different set of evolutionary blueprints, making them the closest thing we will ever find to an alien intelligence thriving right here on planet Earth. To power a decentralized neural network of 500 million neurons and a massive muscular hydrostat system capable of infinite biomechanical contortions, an organism requires an astronomical, uninterrupted supply of metabolic energy and oxygen. In the animal kingdom, the delivery of this vital oxygen is the sole responsibility of the circulatory system. However, the evolutionary path of the octopus forces it to operate in some of the most extreme, hostile, and physically demanding environments on planet Earth. They routinely navigate from warm, sunlit, highly oxygenated surface waters down into the freezing, crushing depths of the abyssal zone, where temperatures plummet and dissolved oxygen levels are dangerously low. To survive this brutal environmental duality, the octopus could not rely on the standard circulatory blueprints found in mammals or fish. Instead, it evolved a wildly complex, highly pressurized, hydraulically extreme cardiovascular architecture centered around the bizarre chemistry of copper and the sheer mechanical force of 3 separate beating hearts. To truly understand this alien cardiovascular system, we must first examine the fluid that pumps through their veins. If you were to puncture the skin of an octopus, you would not see the familiar crimson red associated with vertebrate blood. Instead, the octopus bleeds a brilliant translucent cerulean blue. This radical colour difference is the direct result of a fundamental shift in their microscopic biochemistry. Specifically, the metalloproteins responsible for binding and transporting oxygen molecules. In human beings, and almost all vertebrates, oxygen is transported by a highly efficient protein called hemoglobin. Hemoglobin utilizes iron atoms at its core to bind with oxygen. When iron binds with oxygen, it physically rusts, which is the precise chemical reason why our blood appears red. Furthermore, human hemoglobin is neatly and densely packaged inside specialized disc-shaped containers called red blood cells, which allows our blood to carry massive amounts of oxygen while remaining relatively thin and fluid. The octopus completely discarded this iron-based evolutionary strategy. Instead of hemoglobin, the octopus relies on a massive complex metalloprotein known as hemocyanin. Rather than utilizing iron as the foundational binding agent, hemocyanin is built around atoms of copper. When a copper atom binds with an oxygen molecule from the surrounding seawater, it creates a chemical reaction that reflects blue wavelengths of light, granting the octopus its signature alien blue blood. When the blood is deoxygenated, returning from the muscles and organs, it loses its blue hue and becomes completely clear and transparent. But why did the octopus evolutionarily choose copper over iron? At first glance, this seems like a massive biological downgrade. Hemocyanin is mathematically far less efficient at binding and carrying oxygen than hemoglobin. In warm, oxygen-rich environments, An organism with iron-based blood will heavily outcompete an organism with copper-based blood in terms of pure aerobic endurance. However, the deep ocean is not a warm, oxygen-rich environment. It is a realm of extreme cold and severe hypoxia. In freezing water temperatures, the thermodynamic properties of hemoglobin begin to fail. Cold iron-based hemoglobin binds to oxygen so tightly that it refuses to release the oxygen molecules into the surrounding muscle tissues, effectively suffocating the animal from the inside out despite having full lungs. This is exactly where the copper-based hemocyanin becomes an evolutionary superpower. The molecular structure of hemocyanin is uniquely unaffected by extreme drops in temperature. It maintains its delicate chemical affinity, seamlessly binding to the scarce oxygen molecules in the cold water, and, most importantly, successfully releasing those vital oxygen molecules into the octopus's tissues, even when the surrounding temperature is hovering just above the freezing point of water. By choosing copper over iron, the octopus sacrificed high-performance aerobic endurance in warm water for the ultimate biological insurance policy, the ability to survive and hunt in the freezing hypoxic depths where vertebrate predators simply cannot follow. However, this chemical survival mechanism comes with a devastating, highly dangerous physical trade-off. Unlike human hemoglobin, which is neatly packed into microscopic red blood cells, hemocyanin proteins are far too massive to be contained within cellular walls. Instead, these gigantic complex protein structures float entirely freely, suspended directly within the blood plasma. Because the octopus requires a massive amount of hemocyanin to meet its high metabolic demands, its blood plasma is completely saturated with these giant, free-floating copper proteins. From a purely fluid dynamics perspective, this creates a catastrophic hydraulic problem. The heavy concentration of massive proteins makes the octopus's blood incredibly thick, highly viscous, and remarkably sticky. It has a physical consistency closer to a heavy chilled syrup than to the thin, easily flowing water like blood of a human being. Pumping this thick, heavy, highly viscous blue syrup through a massive, sprawling network of microscopic capillaries poses a tremendous mechanical challenge. The human body requires only one single, powerful heart to pump our relatively thin blood throughout our entire system. If an octopus relied on just one heart, the massive hydraulic resistance created by the sticky, copper-rich blood trying to squeeze through the microscopic blood vessels of its 8 muscular arms and highly dense gills would cause a fatal drop in blood pressure. The singular heart would quickly overheat, fail, and the animal would die of cardiac arrest. To overcome the extreme fluid dynamics of their own viscous blood, evolution engineered a brilliant multi-stage hydraulic pumping system. The octopus possesses 3 completely independent, highly specialized hearts working in absolute synchronization. 2 of these hearts are known as the branchial hearts, or the gill hearts. These are dedicated, localized muscular pumps situated directly at the base of each of the octopus's 2 gills. Their physical job is entirely focused on the respiratory cycle. Deoxygenated, clear blood returning from the vast, decentralized network of the animal's arms and mantle is sluggish and lacking pressure. The 2 branchial hearts draw this depleted blood in and violently pump it directly through the intricate microscopic capillaries of the gills. By applying localized high-pressure force right at the point of respiration, the branchial hearts overcome the severe hydraulic resistance of the gill tissue, forcing the sticky blood to aggressively interface with the surrounding seawater. Here, the copper atoms bind with fresh oxygen, instantly turning the blood a brilliant, vibrant blue. Once the blood is freshly oxygenated, it flows out of the gills and pours into the 3rd central and largest cardiovascular organ, the systemic heart. The systemic heart is a massive, highly muscular, 3-chambered organ consisting of 2 auricles and 1 incredibly powerful central ventricle. This is the primary executive pump of the entire organism. Its sole biological function is to take the freshly oxygenated, highly pressurized blue blood delivered by the branchial hearts and blast it violently throughout the entire body, feeding the massive donut-shaped central brain, the dense optic lobes, and the vast hundreds of millions of decentralized neurons scattered throughout the 8 arms. This 3-heart system is a marvel of biological hydraulic engineering, perfectly solving the problem of high-viscosity copper blood. However, this complex cardiovascular architecture harbours a shocking, fatal flaw, one that fundamentally dictates how the octopus interacts with its physical world. The fatal flaw lies in the anatomical placement of the systemic heart. The systemic heart is located deep within the visceral mass of the octopus, situated immediately next to the primary muscular cavity known as the mantle. The mantle is not just a protective sac, it is the physical engine of the octopus's most explosive defense mechanism, jet propulsion. When an octopus is startled by a predator such as a shark or a moray eel, it rapidly expands its mantle, sucking in a massive volume of seawater. Then, in a fraction of a second, the heavy radial muscles of the mantle violently contract, squeezing the trapped water and blasting it out through a narrow muscular tube called the siphon. This immense, rapid expulsion of water propels the octopus backward through the ocean at terrifying, blinding speeds, allowing it to instantly escape the jaws of death. But the physics of this explosive muscle contraction come at a terrible internal cost. When the massive muscles of the mantle violently clamp down to expel the water, the intense internal physical pressure completely crushes the adjacent systemic heart. The muscle contraction literally squeezes the main heart shut, physically preventing it from beating. This means that every single time an octopus utilizes jet propulsion to swim at high speeds, its primary cardiovascular pump completely stops functioning. The brain and the muscles are immediately cut off from their supply of freshly oxygenated blood. For an incredibly active, highly intelligent creature with massive metabolic demands, shutting down the main blood supply is an unsustainable physiological crisis. As the octopus jets away, it is rapidly suffocating itself from the inside out. It is physically impossible for an octopus to maintain sustained high-speed swimming. After just a few seconds of continuous jet propulsion, the oxygen debt in their tissues reaches critical, agonizing levels. The systemic heart is forcibly stopped. The thick blue blood stagnates in the veins, and the animal experiences total, debilitating muscular failure and absolute exhaustion. This bizarre cardiovascular paradox perfectly explains the everyday behavior of the octopus. This is precisely why, despite possessing the biological capability to swim at lightning speeds, an octopus will spend the vast majority of its life stubbornly crawling along the ocean floor using its 8 arms. Crawling, while slower, is a steady rhythmic motion that does not require the massive continuous contraction of the mantle cavity. While crawling, the systemic heart is completely free to beat normally, maintaining perfect, uninterrupted blood pressure, and ensuring that the complex central brain and the distributed arm brains receive a constant flow of oxygen-rich blue blood. They reserve their incredible swimming speed strictly as an absolute last resort, a desperate panic button pressed only when life and death are separated by milliseconds. They have traded cardiovascular endurance for supreme chemical adaptability, proving once again that their existence is an endless, delicate balancing act of profound biological compromises. The true test of any neural architecture, no matter how bizarre or decentralized it may be, is not merely how it looks under a highly powered electron microscope, but how it actually manifests in the physical, unpredictable world. When we move away from the anatomical dissection tables and observe the living octopus interacting with its environment, we are forced to confront a behavioral psychology that completely shatters the traditional boundaries between vertebrates and invertebrates. For centuries, behavioral science operated on a rigid hierarchical assumption. Complex cognitive functions like anticipatory planning, tool use, facial recognition, and play were the exclusive evolutionary domains of mammals, and perhaps a select few avian species like crows and ravens. Invertebrates, acting essentially as biological automatons, were supposedly driven entirely by hardwired, unthinking instinct. The octopus violently dismantled this scientific dogma. By observing their behaviour, we see the real-world application of their 9 brains, revealing an intellect that is terrifyingly adaptive, profoundly curious, and capable of executing cognitive leaps that scientists previously thought were biologically impossible for a mollusk. One of the most heavily documented and famous displays of octopus cognition is their supernatural ability to solve complex mechanical puzzles, most notably their notorious talent for opening sealed jars to access trapped prey. While this might seem like a simple parlour trick, the underlying neurobiology required to execute this action is staggeringly complex. When an octopus is presented with a glass jar containing a live crab, sealed with a screw-top lid, it cannot rely on evolutionary instinct. There are no screw-top jars in the natural ocean. The animal is facing a completely novel, alien object. To extract the prey, the octopus must engage in rapid, high-level tactile problem-solving. Because the central brain in the head does not possess a strict topographical map of the arms, it cannot simply command the arms to perform a specific twisting motion. Instead, the central brain visually identifies the prey and sends a generalized command to access it. The autonomous arm brains then take over, using their tens of thousands of chemical and mechanical receptors to map the smooth, artificial surface of the glass and the ridged texture of the lid. Through rapid trial and error, the localized arm brains discover that applying rotational pressure to the ridged lid creates movement. The arms coordinate this twisting mechanical force entirely on their own, breaking the seal and unscrewing the cap. But the true scientific marvel occurs after the jar is opened. Once an octopus figures out the mechanical solution to this novel puzzle, it permanently stores this information in its vertical lobe, the neurological equivalent of the mammalian hippocampus dedicated to long-term memory. If you give that same octopus a completely different jar days or weeks later, it will not waste time exploring the glass or trying to pull the lid straight off. It will instantly wrap its tentacles around the lid and apply rotational force. It has genuinely learned, retained, and applied a completely unnatural mechanical concept. Furthermore, their massive optic lobes and complex memory centers grant them a cognitive ability that is incredibly rare in the animal kingdom: the ability to recognize specific human faces. In controlled laboratory environments, researchers conducted experiments where one specific human would consistently feed an octopus, while a different human would consistently poke it with a bristled stick, slightly irritating it. Both humans wore identical uniforms and minimized their body language. Astoundingly, within a very short period, the octopus learned to visually distinguish between the 2 specific human faces. When the friendly feeder approached the tank, the octopus would emerge from its den, swim to the surface, and playfully extend its arms. But when the hostile researcher entered the room, the octopus would instantly retreat to the darkest corner of the tank, flare its chromatophores in a dark, aggressive warning pattern, and utilize its siphon to blast a high-pressure jet of water directly at the researcher's face. This proves that their decentralized intelligence is not just a reactive survival engine. It is capable of complex associative learning, emotional memory retention, and targeted, highly personalized physical retaliation. However, the absolute pinnacle of their behavioral psychology, and the discovery that truly forced scientists to rewrite the textbooks on invertebrate cognition, was documented in the year 2009 off the coast of Indonesia. Marine biologists were observing a species known as the veined octopus, scientifically named Amphiooctopus marginatus, and witnessed a behavior so sophisticated it sent shockwaves through the scientific community. They observed these octopuses actively searching the ocean floor for discarded, hollowed-out coconut shells halves. When an octopus found a suitable shell, it would meticulously excavate the mud and sand out of the interior. But it did not just use the shell as a temporary hiding spot like a standard hermit crab. Instead, the octopus would stack multiple coconut shell halves together, wrap its body entirely around them, and physically carry this heavy, clunky cargo across the barren, exposed ocean floor. To carry this massive object, the octopus had to contort its body into a bizarre, highly unnatural posture, essentially walking on tiptoes using only the very rigid tips of a few tentacles, a movement biologists dubbed stilt walking. Carrying these heavy shells makes the octopus incredibly slow, highly visible and extremely vulnerable to predators. It requires a massive expenditure of metabolic energy and is biomechanically exhausting. So why would they do it? They carry the shells so that later, when they are travelling across an open stretch of sand with absolutely no natural cover, they can instantly drop the shells, climb inside, and pull the two halves completely shut, creating a perfect, impenetrable spherical fortress. From a cognitive psychology perspective, this is a monumental breakthrough. This is not merely utilising an object that happens to be nearby. This is absolute, undeniable evidence of anticipatory tool use. To engage in this behaviour, the octopus must possess a highly advanced cognitive concept known as mental time travel. The central brain must be able to project itself into a hypothetical future scenario. It must consciously realise, I am currently safe, but in the near future, I will be in a dangerous, exposed environment. Therefore, I must endure a severe physical penalty right now to carry this tool so that I will have it when I inevitably need it later. Before this discovery, scientists believed that this level of complex forward planning and abstract tool retention was strictly limited to humans, higher primates, and select corvids. The fact that a soft-bodied mollusk, operating on a decentralized network of ganglia, is capable of anticipatory tool use is one of the most profound biological discoveries of the 21st century. It proves that deep, analytical thought is not a mammalian monopoly. This high-level intelligence also manifests in behaviours that have absolutely zero immediate survival value, most notably the capacity for genuine play. In biology, play is considered a luxury of the intelligent. If an organism is merely a biological machine running on basic survival scripts, it will conserve its energy for hunting, mating, or hiding. But octopuses frequently exhibit a massive cognitive surplus. In aquariums, they have been observed taking floating plastic pill bottles and intentionally positioning them over the water jets in their tanks, watching the bottles get blasted across the surface. They will then swim over, retrieve the bottle, bring it back to the jet and do it again, repeating this cycle for hours. They are not trying to eat the bottle, and they are not defending their territory. They are actively seeking mental stimulation. Their complex neural pathways demand constant engagement, proving that their brains require active, continuous interaction with the physical physics of their environment to remain healthy, much like the brain of a human child. Finally, to truly understand the depth of their psychological complexity, we must observe what happens when this massive neural network shuts down for the night. Octopuses experience complex sleep cycles that heavily mirror the neurological patterns found in mammals. Their sleep is fundamentally divided into two distinct, alternating phases: quiet sleep and active sleep. During quiet sleep, the octopus becomes completely motionless. Its breathing slows down significantly. Its pupils narrow into tiny slits, and its skin turns pale and completely smooth. It is a period of deep physiological rest, but roughly every 30 to 40 minutes, the resting octopus violently shifts into active sleep. While the animal remains completely unconscious, its body erupts into a frenzy of automated activity. Its closed eyes dart around wildly beneath the skin, its arms spontaneously twitch, and the suckers aggressively contract and release as if grasping invisible objects. But the most spectacular aspect of active sleep is the skin. The chromatophores and iridophores begin to flash violently. cycling through incredibly complex high-definition patterns of colour and texture. 1 second, the sleeping octopus is flashing the dark red, spiky texture it uses to mimic venomous algae, and the next second it shifts to the pale, smooth pattern it uses to ghost across white sand. Neurologists analysing this active sleep phase have come to a staggering conclusion. The shifting colours and muscular twitches are not random biological static. This sleeping octopus is displaying the exact precise physiological sequences of camouflage and movement that it utilises when hunting prey or hiding from predators during its waking hours. The most plausible scientific explanation is that the octopus is experiencing the invertebrate equivalent of rapid eye movement sleep. Just like a human brain replays the events of the day during dream sequences to strengthen synaptic connections and consolidate long-term memories, the central brain of the octopus is actively replaying its episodic memories. It is mentally reliving its successful hunts and terrifying escapes, and because its skin is directly wired into its neural network, its body acts as an external high-definition monitor, broadcasting its internal dreams for the world to see. When you combine their capacity for associative learning, their ability to recognize individual faces, their mastery of anticipatory tool use, their desire for unstructured play, and the vivid, memory-consolidating nature of their sleep cycles, the behavioral profile of the octopus ceases to look like that of a simple sea creature. We are looking at an animal that possesses a rich, complex inner life, an organism that thinks, plans, remembers, and dreams. They are a profound reminder that the dark depths of the ocean are not just filled with biological survival machines. They are inhabited by deeply conscious, intelligent minds, observing the world through massive U-shaped pupils and processing reality in a way we're only just beginning to comprehend. When you look at the entirety of the octopus's biological portfolio, its massive, deeply complex genome, its unparalleled ability to edit its own ribonucleic acid on the fly, its distributed intelligence across 500 million neurons, and its capacity for associative learning and anticipatory tool use, you're looking at one of the most sophisticated, high-performance biological machines ever produced by planet Earth. In the broader context of evolutionary biology, there is a very strict, almost unbreakable mathematical correlation between high-level intelligence and long physiological lifespans. Building a massive brain requires an astronomical investment of metabolic energy, time, and caloric resources. Consequently, highly intelligent animals like humans, elephants, chimpanzees, and certain species of whales and parrots are evolutionarily designed to live for decades, allowing them to maximize the return on that immense biological investment. But the octopus brutally shatters this foundational biological rule. Despite possessing a mammalian level of intelligence and a body capable of shape-shifting optical illusions, the vast majority of octopus species possess a maximum lifespan of just 1 to 2 years. Some smaller species live for only 6 months. This creates the ultimate evolutionary paradox. Why would nature invest such a staggering amount of genetic and metabolic capital to build a creature that is fundamentally a biological supercar, only to program that car to violently self-destruct the moment it drives off the assembly line? The answer lies in their reproductive biology, and it reveals an endocrine system mechanism that is as fascinating as it is deeply tragic. For the octopus, reproduction is not a mere phase of life. It is an absolute non-negotiable death sentence. They are what biologists classify as simulparous organisms, meaning they reproduce only a single time in their entire lives, and that singular act triggers a catastrophic, irreversible cascade of biological self-destruction known as senescence. The ticking of this fatal biological clock is initiated the moment they mate. The mating process itself is a highly precarious, socially distant affair, primarily because octopuses are notoriously cannibalistic. A male octopus must approach a larger, highly aggressive female with extreme caution. Instead of a traditional biological embrace, the male utilizes a highly specialized modified third arm known as a hectocotylus. This specialized arm contains a deep groove and is designed to physically hold and transfer packets of sperm, known as spermatophores, directly into the mantle cavity of the female. Once this incredibly tense, high-stakes biological transfer is complete, the male immediately retreats. His evolutionary purpose has been entirely fulfilled. Almost immediately after mating, the male's digestive system begins to shut down. He stops hunting, his decentralized brain begins to deteriorate. He wanders aimlessly into the open water, and within a matter of weeks he dies of starvation, or is easily picked off by predators. But the physiological journey of the female is infinitely more complex, drawn out, and scientifically horrifying. After mating, the female searches for a secure, deeply hidden crevice in the rocks. She will lay tens of thousands, sometimes up to hundreds of thousands, of microscopic translucent eggs, meticulously braiding them together and hanging them from the ceiling of her den like strands of crystal pearls. Once the last egg is laid, a highly specialized neurological switch is flipped deep within a central donut-shaped brain. For decades, marine biologists were utterly mystified by what exactly caused the mother octopus to die. Was it sheer exhaustion from producing so many eggs? Was it a lack of food? In the late 1970s, a massive scientific breakthrough occurred. Researchers discovered that the entire process of senescence is strictly controlled by a tiny pair of organs situated directly between the octopus's massive eyes, known as the optic glands. The optic gland is the invertebrate equivalent of the human pituitary gland, acting as the absolute master control center for the animal's endocrine system. To prove the function of this gland, a scientist named Jerome Wodinsky performed a radical surgical experiment. He took a female octopus that had just laid her eggs and was beginning to starve herself, and he surgically severed the neural connections to her optic glands. The results were immediate and shocking. The female completely abandoned her eggs, swam out of her den, began aggressively hunting and eating again, and effectively doubled her natural lifespan. This experiment proved beyond a shadow of a doubt that the death of the octopus is not accidental. It is actively, chemically forced by her own body. But it was not until the year 2022 that molecular biologists finally decoded the exact chemical weaponry the optic gland uses to execute the animal. By analysing the glandular secretions of female octopuses during the brooding phase, scientists discovered a massive fatal shift in the animal's cholesterol metabolism. When an octopus lays her eggs, the optic gland dramatically ramps up the production of specific steroid hormones, including molecules deeply related to pregnenolone and progesterone. Simultaneously, it completely halts the synthesis of standard cholesterol. In a human being, a healthy balance of cholesterol is vital for maintaining the structural integrity of cell membranes and the proper function of the nervous system. But in the brooding mother octopus, the optic gland intentionally floods her circulatory system with highly toxic, unrefined precursors to cholesterol. This sudden, violent hormonal storm systemically poisons the animal from the inside out. The physical manifestation of this endocrine hijacking is brutal to witness. The mother completely ceases to hunt. Her digestive tract effectively atrophies and shuts down. For months, she dedicates 100% of her failing metabolic energy to her eggs. She constantly uses her siphon to gently blow fresh, oxygenated seawater over the egg strands to prevent fungal and bacterial infections, and she fiercely defends the den against crabs and predatory fish. As the starvation and hormonal toxicity reach their absolute peak, the animal enters a phase of rapid macroscopic cellular decay. She begins to lose colour, her skin turning a sickly, ghostly white. Her muscles lose their tone, and she often enters a phase of autophagy, literally beginning to tear off her own skin and eat the tips of her own tentacles in a display of programmed neurological malfunction. She clings to life just long enough to see the eggs hatch. The moment the thousands of microscopic baby octopuses break free from their casings and float up toward the ocean surface, the mother's biological imperative is complete, and she dies on the floor of her den. But why would evolution, a system driven by the relentless pursuit of survival, select for such a cruel and biologically expensive design? Why program the optic gland to execute the most intelligent invertebrate on Earth just as it reaches full maturity? The scientific consensus points to three brilliant intersecting evolutionary justifications. The first justification is the prevention of generational cannibalism. Octopuses are voracious, highly efficient, opportunistic apex predators of the micro-ecosystem. They will eat anything they can physically overpower, including other octopuses. If a massive, highly experienced mother octopus were to survive the hatching of her eggs, and she was starving after months of brooding, the first thing she would encounter upon leaving her den would be a cloud of 100,000 microscopic, highly nutritious baby octopuses. To prevent the mother and the roaming males from instantly decimating the next generation, natural selection chemically removes the parents from the ecosystem before the children are even born. The babies hatch into an ocean completely devoid of the one predator that knows exactly how they think and where they hide. The second evolutionary justification relates directly to their unique genetic architecture, specifically the ribonucleic acid editing we discussed earlier. Because the octopus relies so heavily on editing its ribonucleic acid to adapt to immediate environmental changes, its foundational deoxyribonucleic acid mutates at an incredibly slow rate. In the evolutionary arms race, slow genetic mutation is a severe disadvantage when competing against fast-evolving viruses, parasites, and predatory fish. To compensate for their stagnant deoxyribonucleic acid, the octopus lineage employs a strategy of hyper-rapid generational turnover. By dying after 1 year and producing hundreds of thousands of offspring, they force the evolutionary dice to be rolled millions of times in a very short biological window. This massive volume of rapid reproduction mathematically guarantees that at least a tiny fraction of the offspring will possess the necessary genetic variations to survive emerging ecological threats. They sacrifice the longevity of the individual to ensure the rapid dynamic genetic survival of the entire species. The third, and perhaps most profound evolutionary justification, is the concept of an ecological safety valve. You must consider the sheer, terrifying potential of an animal equipped with 9 brains, the ability to turn invisible, a fluid body completely unbound by bones, the capacity to use tools, and an incredibly aggressive predatory instinct. If an octopus possessed the 80-year lifespan of a human being, allowing it to accumulate decades of complex memories, perfect its hunting strategies, and grow continuously without the constraints of a shell, it would become an unstoppable ecological nightmare. They would systematically eradicate every crustacean and small fish in their territory. They would completely dominate the coastal food webs, inevitably collapsing the very ecosystems they rely upon, which would ultimately lead to their own starvation and extinction. Their spectacularly short lifespan is the universe's ultimate balancing act. Their rapid death is the heavy mandatory tax they must pay for possessing such radical, alien-like biological superpowers. They are granted a fleeting, brilliant existence, given just enough time to master the physics of their environment, solve the puzzles of the reef, and pass on their genetic blueprint before a chemical switch in their brain quietly extinguishes the light. The tragedy of the octopus is not that it dies so soon, but that it burns so incredibly bright while it is here, representing a totally alternate pathway of conscious life that visits the Earth for only a brief spectacular moment. The profound, alien-like biology of the octopus is not just a subject of academic curiosity for marine biologists. It has become one of the most highly sought-after blueprints in the modern fields of engineering, material science, and robotics. For decades, human engineering has been fundamentally constrained by the paradigm of rigid materials. We build robots, vehicles, and tools using hard metals, rigid plastics, and jointed skeletal structures. But when engineers look at the octopus, they see a highly successful biomechanical organism that completely threw away the concept of rigid joints hundreds of millions of years ago. By studying how the octopus moves, feels, and hides, scientists are now pioneering a revolutionary new field known as soft robotics and biomimetic materials, attempting to replicate the octopus's superpowers in artificial systems. The most direct and immediate application of octopus biology is in the rapidly expanding field of soft robotics. Traditional rigid robots are incredibly powerful and precise, but they are inherently dangerous to humans and terrible at navigating unpredictable, unstructured environments. A rigid robotic arm designed to weld car parts cannot safely reach into a collapsed building to gently extract a trapped survivor, nor can it safely navigate the delicate, squishy environment of the human body during surgery. The octopus arm, however, is the perfect biological solution to these problems. Biomedical engineers have spent years meticulously analysing the muscular hydrostat architecture of the octopus tentacle. By mimicking the dense, intersecting arrays of longitudinal and transverse muscle fibres, engineers are constructing artificial boneless robotic arms using highly flexible, biocompatible silicone polymers. Instead of using electrical motors and metal hinges, these soft robotic arms are driven by complex pneumatic and hydraulic networks. By selectively inflating and deflating microscopic fluid chambers embedded within the silicone, engineers can force the artificial arm to bend, stretch, twist, and grip with the exact same infinite degrees of freedom possessed by a living octopus. These biomimetic soft robots are currently being developed for highly delicate surgical procedures. A rigid surgical tool can only move in straight lines, but a soft, octopus-inspired robotic catheter can snake its way through the complex, winding blood vessels of the human cardiovascular system, or navigate the delicate tissues of the brain without causing internal lacerations. Furthermore, these soft robotic arms are being equipped with artificial suction cups. Engineers have reverse-engineered the mechanical pressure dynamics of the octopus sucker, creating artificial grippers capable of lifting heavy, irregularly shaped objects, ranging from delicate raw eggs in a food processing plant to heavy, submerged debris in deep-sea exploration, all without utilizing a single rigid hinge. But the physical mechanics of the arm are only half of the equation. To truly replicate the octopus, these soft robots need to be smart, and this is where the concept of distributed, decentralized intelligence is revolutionizing computer science. As discussed previously, the central brain of the octopus does not micromanage the arms. It issues high-level commands, and the localized arm brains execute the complex mathematics. Robotics engineers are adopting this exact biological architecture to solve the massive computational bottlenecks in robotic control systems. In traditional robotics, a central computer processor must constantly calculate the exact coordinates of every single joint and motor. For a robot with dozens of joints operating in a complex environment, this requires massive processing power and often results in sluggish, delayed movements. By mimicking the octopus's localized ganglia, engineers are developing decentralized robotic networks. They are embedding microprocessors and sensory feedback loops directly into the limbs of the soft robots. This allows the robotic arm to locally process tactile data and make micro-adjustments to its grip or trajectory without constantly sending data back to the central processor. This biomimetic approach, known as morphological computation, drastically reduces the data load on the main computer, allowing the robot to react to unpredictable environments with the same fluid, real-time speed as a living cephalopod. Beyond robotics, the octopus's mastery of optical physics is driving a massive revolution in materials science, specifically in the development of active camouflage and smart fabrics. The military and the aerospace industry have poured millions of dollars into understanding exactly how the octopus's chromatophores and iridophores function. Their goal is to move away from static, printed camouflage patterns and develop dynamic materials that can actively change color and texture to match any environment in real time. Material scientists have successfully created artificial chromatophores, using dielectric elastomers. These are ultra-thin, flexible polymer membranes filled with dark, carbon-based pigments. By sandwiching these pigment-filled membranes between flexible electrical grids, scientists can mimic the neurological control of the octopus. When an electrical voltage is applied to the grid, the polymer expands, spreading the pigment out and turning the material dark. When the voltage is removed, the polymer snaps back and the material becomes transparent. By layering thousands of these microscopic artificial chromatophores, engineers are developing thin, flexible sheets of material that can rapidly shift between complex patterns of black, white, and grey. Furthermore, physicists are working to replicate the structural colour manipulation of the octopus's iridophores. By using advanced nanotechnology, they're engineering synthetic protein arrays and photonic crystals that can actively tune their physical spacing in response to electrical or chemical stimuli. This allows the artificial material to actively reflect specific wavelengths of light, instantly shifting from a deep blue to a vibrant green, exactly like the skin of a living octopus. When you combine artificial chromatophores, synthetic iridophores, and highly sensitive decentralized light sensors mimicking the opsin proteins found in octopus skin, you get the holy grail of materials science, adaptive, biomimetic skin. While still in the experimental phases, these technologies promise a future where military vehicles can literally disappear into the background, where civilian clothing can actively regulate temperature by shifting its thermal reflectivity, and where the very walls of our buildings can dynamically change colour and texture in response to ambient light. We are systematically dismantling the biological magic of the octopus, translating its ancient evolutionary secrets into mathematical equations and synthetic polymers, proving that the most advanced engineering blueprint on the planet is currently swimming in the ocean, covered in suckers and completely lacking a spine. When astrophysicists and astrobiologists at NASA look out into the cosmos searching for extraterrestrial life, they are no longer just looking for microbes on Mars. They are turning their immense telescopes toward the icy moons of the outer solar system, specifically Jupiter's moon Europa and Saturn's moon Enceladus. Deep beneath the miles of frozen ice crust on these moons, planetary scientists have confirmed the existence of massive global liquid water oceans, kept warm by the tidal friction generated by their massive host planets. These subterranean alien oceans are currently considered the most promising environments in our entire solar system to harbour complex multicellular life. However, designing a robotic probe to search for life in a pitch-black, freezing, highly pressurised extraterrestrial ocean presents a massive conceptual problem. What exactly are we looking for? If life exists down there, it certainly will not look like a human being, and it likely will not operate on vertebrate architecture. This is precisely why astrobiologists consider the octopus to be the ultimate living proxy for alien life. The octopus provides a perfect, functioning biological model of how high-level intelligence and complex behaviour can evolve entirely underwater in environments of extreme pressure and profound darkness without ever needing fire, dry land, or a rigid skeletal structure. NASA studies the octopus because it proves that a decentralized nervous system is a highly successful evolutionary strategy in aquatic environments. If a probe lands on Europa and encounters a complex organism, it is highly probable that the creature will not possess a centralized brain encased in a skull. Instead, it is much more likely to resemble the octopus, a fluid, shape-shifting entity with processing power distributed throughout its limbs, allowing it to navigate a zero-gravity aquatic environment with infinite degrees of freedom. Furthermore, their ability to edit their own ribonucleic acid to survive freezing temperatures provides astrobiologists with a plausible biochemical blueprint for how life could rapidly adapt to the extreme, fluctuating thermal vents at the bottom of an alien ocean. By analysing the genome, the neurobiology, and the behavioural psychology of the octopus, NASA is essentially studying a successful alien prototype. The octopus is our single best piece of empirical evidence that the universe has more than one way to build a conscious mind, offering us a vital glimpse into what might be waiting for us in the dark, icy waters of deep space. The octopus is not an invader from the cosmos. Its massive genome, its ability to rewrite its own ribonucleic acid on the fly, its decentralized 9-brain network, and its mesmerizing optical physics are not the products of an alien meteor. They are the ultimate testament to the raw, unyielding power of evolution right here on planet Earth. The octopus proves that our world is far more alien and wonderful than we often realize. It demonstrates that the universe has more than one way to build a highly intelligent, conscious mind using a completely different set of biological rules. Earth's oceans are still hiding secrets that challenge our fundamental understanding of life itself. If you found this deep dive into the weird and wonderful science of the natural world fascinating, please hit the like button and subscribe to the channel. Leave a comment below sharing your thoughts, and let us know what scientific mystery we should explore next. Thank you for watching.